Hi everyone, welcome to my new video. In this video, we talking about the mitochondria. In previous video, we talking about the history of mitochondria, size, number, and position of mitochondria. Okay, in some form of their structure. Right now, in this video, we talking about the structure of the mitochondria. Okay, the microscope shows the mitochondria as the vesicle bounded by an envelope. Okay, it means here a vesicle present, and this vesicle bounded by the one envelope of 2-unit membrane and filled with the fluid matrix. It means here the unit 2-unit membrane present here. It means inner membrane and the outer membrane, and this inner membrane is filled with the fluid matrix right now, and this is the structure of the mitochondria, and there are different parts present here. Okay, then membrane. Both the inner and outer mitochondrial membrane resemble the plasma membrane. Okay, in molecular structure. It means this both membrane are same like that of the plasma membrane in molecular structure. Okay, and each of them is 60 to 70 angstrom. Trilaminar and composed of two layers of phospholipid molecule and sandwiched between the two layers of protein molecule. You all know that this plasma membrane contains from the protein, lipid and protein structure. It means trilaminar structure. Okay, and this phospholipid molecule are sandwiched between these two layers of protein molecule. Okay, it means lipid is sandwiched between the two layers of protein molecule right now, and the outer and inner membrane are separated from each other by the narrow space, and this narrow space is called as the intermembrane space or the outer chamber or perimitochondrial space right now. It means this intermembrane space, this is here, you can see that inner and outer membrane present, and here the outer chamber present, this space present between these two layers, that is outer and inner membrane right now. Then going toward the outer membrane, this outer membrane is a smooth permeable to most small molecules, having a transmembrane channel formed by the protein porine. It consists of about 50% lipid, including large amount of the cholesterol, and it contains some enzymes but in porine protein right now. It means this outer membrane have the large number of lipids present here and very less amount of the protein present here. Okay, then going toward the inner membrane, this inner membrane is selectively permeable and regulate the movement of material into and out of the mitochondria. Okay, it means outer membrane is a smooth permeable, but inner membrane is a selectively permeable right now, and it is a rich in enzyme and carrier protein permeates. Okay, and it has a very high protein or lipid ratio, about 4:1. Okay, and it lacks cholesterol. It means here the protein, a large amount of the proteins present here in the inner membrane. Okay, this is about the outer and inner membrane. Then going toward the matrix. Okay, the space between the cristae called the inner chamber is filled with the gel like material termed as the mitochondrial matrix. It means the inner membrane, the fluidy material is present, and this fluidy material is called as the matrix. It in this matrix, proteins present, lipids present, some ribosomes present, RNA or the 2 DNA molecule or certain fibrils, crystals and dense granules present in this matrix. Okay, and this matrix is very important here because all the material present in this matrix here. Okay, then in these then next one point is the cristae. The inner mitochondrial membrane bear plant-like enfoldings, plate-like enfolding, not plant-like, sorry, plate-like enfolding here. Okay, it means the inner membrane is a folding-like structure present here, plate-like enfolding, that is called as a cristae, and they extend inward to varying degrees and may fuse with those from the opposite side, dividing the mitochondria into two compartments. Okay, it means this folding in the inner side into the matrix, this is called as the cristae. Okay, here in this diagram we can see that in this inner folding of the membrane, that is called as the cristae. They are arranged in the characteristic manner in different cell. Normally they run in right angle to long axis and the rod shaped mitochondria. Right now, in cell of the proximal part of the kidney tubule, the cristae are longitudinal fold parallel to the long axis of the mitochondria. Okay, and the in heart muscle cell, cristae are zigzag. Okay, they also vary in number. This cristae also have the vary in number, and the active cell may have the numerous cristae like that of the mitochondria, whereas the inactive cells may have the only few. Okay, same like that of the mitochondria also have the in some cell there are more in number, in some cell they are less in number. Why? Because they are depend on the cell type, cell activity. Okay, here also like that of the mitochondria. Then oxysomes. The inner mitochondrial membrane bear minute regularly spaced particles. It means in inner mitochondrial membrane the very small particles present here, and this is called as the inner subunit, or that is called as the elementary particle or oxysomes. Okay, this oxysome also called as the elementary particle, and oxysome consists of the three parts: rounded headpiece or F1 subunit joined by the short stalk to the base piece, or F0 subunit located in the inner membrane. It means here the first one is what? Head piece present, okay, or also called as the F1 subunit, then short stalk present here, and then base piece present here. It means here three parts present of the oxysome or the elementary particle. Okay, here we can see that detailed structure of the crista. It means single this enfolding is called as the crista or an oxysome. In this structure we can see that here the matrix of inner membrane present here, inner membrane space in the outer chamber present here the rounded head present, that is F1 subunit present, then stalk present, and then the base piece present here. Okay, in this diagram you clearly see here that is the head, stalk, and base piece present here. Okay, then biogenesis of mitochondria. The formation of new mitochondria has been explained with the following hypothesis. The origin of mitochondria is take place from the de novo synthesis, origin from the membrane. Okay, you all know that lysosome origin take place from the ER or the Golgi complex. Okay, and these mitochondrial origin take place from the de novo synthesis or origin from the membrane. Okay, then origin from the development from the micro bodies or the prokaryotic origin also, and the replication origin also take place here. Okay, then going toward the function of mitochondria. Cell respiration take place in the mitochondria, and so they are known as the powerhouse of the cell. You all know that mitochondria is called as the powerhouse of the cell. Why? Because cell respiration take place in the mitochondria. Then mitochondria provided intermediate for the synthesis of important biomolecules such as chlorophyll, cytochromes, steroids, etc. Okay. Then some amino acids are also formed in the mitochondria. Okay. Then mitochondria actively accumulate calcium ion as calcium phosphate precipitate. They regulate the calcium ion concentration in the cytoplasm by storing and releasing. Okay, it means this mitochondria actively accumulate calcium ion, okay, as a calcium phosphate precipitate. Okay, and the calcium ions regulate numerous biochemical activities in the cell. Okay, this is all about the structure of mitochondria. Okay, then biogenesis of mitochondria, then function of the mitochondria. Okay, and these all are the references used during the preparation, at the time of preparation. Okay, thank you very much.